Greetings and thanks for being here. My name is Kejun Coco, major in biochemistry and molecular biology. My IS project investigates the logic puzzle of traditional Chinese medicine by deciphering the cooperative protective effect of two chemicals, Hyperoside and Artemisinin, against oxidative damage. My passion has been to contribute to the understanding of traditional Chinese medicine and natural remedy through the lenses of biochemistry. For background, we human beings are under constant threat from reactive oxygen species, ROS, the unstable molecules that contain oxygen. They can be created by electromagnetic radiation from the environment, such as pollution, as well as cellular metabolism, such as respiration. High level of ROS correlate with aging and diseases such as diabetes, cardiovascular diseases, and even cancer. Fortunately, antioxidants are capable of preventing oxidative stress in our body by neutralizing ROS free radicals. Those antioxidants can be found in natural medical remedies that are utilized in traditional Chinese medicine. Clinical application of Chinese herbal medicine have been documented for centuries. Discovered recipes usually have different combinations of herbs for different symptoms or diseases. In my screening through the documented recipes, I found that Artemisinin annua and Lonicera japonica plants are frequently used together for the purpose of detoxification and heat clearing, which is similar to the consequence of curing from oxidative stress. However, like many Chinese herbal medicine treatments, the underlying bioactive mechanism of the treatment has not been completely elucidated. Medical herb and their bioactive ingredients have been studied in order to link specific compounds with their biomedical functions. For the AALJ herb pair of interests, Artemisinin was discovered and identified as the bioactive ingredient of Artemisinin annua by Yo-Yo Tu in the 1970s. This study of Artemisinin was a Nobel Prize awarded anti-malarious treatment. From the other plant of herb pair, Lonicera japonica, one of the bioactive ingredients is Hyperoside, which was proven to be able to enhance the antibiotic effect of another natural remedy. Interestingly, both Artemisinin and Hyperoside were found involved in protection against oxidative damage, though the detailed mechanisms still remain largely unknown. Those studies that identified and study a single herb and bioactive ingredients provided important insights, but rarely have research considered the outcome of using combination of active ingredients. It is important to consider how combining more than one ingredient create different effects, since many Chinese herbal medicines are mixture with different plants. This may enhance effectiveness or to reduce side effects. Therefore, my specific aim is to test the cooperative enhancement effect of ART and Hyperoside with the tool of antioxidant evaluation. The basic approach will be to test Artemisinin and Hyperoside with baker's yeast. Evaluation will be made based on the endurance of yeast within the oxidative stress condition. I hypothesize that when used together, Artemisinin and Hyperoside will have the enhanced antioxidant effect on yeast. I would expect it to have significant increase in yeast survival rate when compared to the treatment of only either one of them. Shown here is the experimental design flow. Yeast was treated with one or both Artemisinin and Hyperoside antioxidants for 24 hours before being exposed to one of the oxidants of trace. Zero dilution was followed by spotting of culture on solid YPD plate. This allowed counting for the number of colonies by naked eyes. The practical question in lab now is to control the two important variables, the concentration of the DMSO solvent and the concentration of applied antioxidants as circled in red. For the concentration of DMSO solvent, 3.5% concentration by volume was selected based on the evaluation follow the similar step as shown in the previously presented experimental flow, but without oxidant or antioxidant treatment. Here, exposure to higher concentration of DMSO, specifically when higher than 3.5%, for 24 hours induce inhibition of cell viability. For the second question, to select for the concentration of antioxidants, each cell viability was measured with treatment of either of ART or Hyperoside for 24 hours without applying oxidants. The results showed that antioxidants showed no significant toxicity until the concentration exceeds 200 micromolar, as there is a decrease of survival rate in E cell in both tests for artemisinin and Hyperoside. Therefore, I selected 200 micromolar as the concentration of ART and Hyperoside for the next experimental evaluation. This final evaluation shows synergistic effect in protection against oxidative damage in two forms. First, according to the result, when exposed to hydrogen peroxide, the survival rate was surprisingly as high as 62.5% with treatment of both ART and HYPE. But the same oxidative damage was not effectively prevented by either one of them, though the enhanced antioxidant effect did not persist for a long time, as we see the survival rate drop rapidly to 4.3% and lower after 24 hours of exposure. When the yeast culture was treated with both artemisinin and Hyperoside to prevent carbon tetrachloride-induced damage, as shown in the figure below, the survival rate was persistently higher than 60% after 1, 24, and 48 hours of exposure. The protective effect lasted longer, though it was not the most effective as compared to the Hyperoside treatment after 1 hour only. Taken together, this project indicated that the natural remedies Hyperoside and artemisinin serve as antioxidant protectors for yeast in response to the stimulation of oxidative stress, and there was cooperative enhancement in antioxidant protection effect when both are used. Such an effect existed in two forms with different sources of oxidative damage. Broad and major future directions are possible. First, it is worthy to examine what is the response mechanism in yeast that allowed Hyperoside and artemisinin to enhance protection in oxidative damage. Second, we can further explore the treatments in scripts and their functions. This might allow for determination of optimal ratio for each bioactive ingredient, induce more effective formulation, and possibly refine Chinese herbal medicine treatment with reduced side effect, more easily standardized system. I would like to thank Yali Wu, my mother and a passionate pharmacist who research in traditional Chinese herbal medicine, and Jinbiao Liu, my chemist's father, and all my family members. I am grateful for my advisor, Dr. Sarah Martin, the second reader, Dr. James West, and all professors in the BCMB program in the College of Wooster for their wisdom. Thanks also to my friends and classmates at the College of Wooster for their support at this time. A special thanks goes also to my American host families, the Hemmings and the McCollums, for their spiritual, mental, and physical support. This study was funded by Copeland Fund from College of Wooster.